Hello everyone, I'm Dr. Deepak. I'm from the ICMR's National Institute for Research in Reproductive and Child Health, and my lab is interested in understanding what are the fundamental mechanisms that drive pregnancy initiation, how the embryo and the maternal endometrium interact, leading to the implantation of the embryo and early presentation, and how things go wrong in these interactions to lead to infertility and pregnancy-associated birth complications. Very recently, we have discovered the fundamental molecular driver of the process of embryo implantation, where we discovered how epithelial to mesenchymal transition in the endometrial epithelium aid in embryo implantation. And this work can be read if you scan this QR code. And also, we have recently reported how inflammation in the maternal endometrium is controlled, leading to successful implantation. And in the switch of inflammation is disrupted, it can lead to defective placentation and early pregnancy losses. And this work can be accessed through this QR code. In addition, we are also interested in a lot of translational research, where our work on genetics of male infertility and use of our artificial intelligence-based software to predict the outcomes of IVF in infertile males have been commercialized. We are also developing organ-on-chip and organoid models of female reproductive tract, and in collaboration with Professor Abhijit Majumdar, we have developed placenta-on-chip, and he has discussed about it. For today's talk, I'm going to discuss about how infections are associated with preterm births. And using this information that we have now gathered, we are in the process of developing dissection of common infections for predicting preterm births. And the work that I'm going to talk to you today has been largely done by my collaborative graduate student, Abhishek Singh, and now spearheaded by my technical staff, Sarika. And Abhishek was supported by Karishma, Neerja, and Atar, and I'm eternally grateful to them. Now, India is a very populous country, and we have a preterm birth rate of around 13%, and which means that almost 8 babies are born preterm every minute, and therefore, by the time I finish this talk, nearly 200 babies will be born premature. And this is a very large number, and of course, not a surprise that India accounts for 26% of the world's preterm birth burden. Now, preterm birth is defined as extreme preterm, which are the babies which are born before 28 weeks of gestation, very early preterms, which are born between 28 to 32 weeks of gestation, and moderate to late preterm, that is after 32 weeks. And this early delivery is a problem because preterm birth is the highest risk factor for infant mortality, and India is on the leaderboard of infant mortality. In addition, it is a cause of neonatal infections, growth retardation, and multiple neurodevelopment disorders. Therefore, identifying what are the causes of preterm birth is crucial to develop modalities for diagnosing it and preventing, and therefore, and eventually treating and reducing the burden of preterm birth in India. Now, studies have shown that preterm births, there are multiple maternal factors which are associated with preterm birth, and often infection, especially bacterial infections, is a common cause. Often studies done earlier have shown that vaginal colonization with an organism called as Croupi streptococcus, or in short GBS, is strongly associated with preterm births. And our interest in preterm birth and GBS started with a collaborative study with Professor Anirban Banerjee at IIT Bombay, where he had first reported that the GBS bacteria produces microvesicles. These are tiny bud-like structures which are released by the bacteria, and they contain a lot of virulence factors. If we take these microvesicles and instill them in the vagina of mice, we showed that they could transverse anterogradely upwards and sit inside the female reproductive tract. And if we now give these microvesicles to GBS-derived microvesicles to pregnant mice, they cause histological chorioamnionitis, which is strongly associated with preterm birth. And most of these animals which are administered with GBS microvesicles delivered preterm, and a proportion of them also at intrauterine fetal death. And this established that mechanistically how colonization of this pathogenic bacteria only in the vagina. The organism need not enter greatly transport, but these microvesicles can go up into the female reproductive tract, cause inflammation, cause collagen degradation, leading to premature rupture of membranes as well as preterm births. And the studies can be accessed if you scan these QR codes. And this was a very exciting time because we were showing now mechanistically how group B streptococcus, along with its virulence factors, can cause preterm birth. Now we are looking at what's the incidence and what are the different causes of preterm births in India. And where many studies had shown that the vaginal microbiome is strongly associated with preterm birth. Now we know different tissues, especially the gut, is full of different microbes, and any disruption in this microbial homeostasis can lead to pathogenicity. And in the context of preterm birth, several studies from the Caucasian population had shown that during pregnancy, there's a shift in the microbiome of the vagina. And if this shift is disrupted, it is associated with preterm birth. So the question we asked was that is vaginal dysbiosis is a cause of preterm birth in Indian women? At that point, Abhishek had joined me, and he had started the study initially at MGM Hospital, Women's Hospital at Kalamboli, Navi Mumbai. And eventually, we also did a cross-sectional study at the Navroz Jivadia Maternity Hospital. Both of these hospitals cater to the rural and the semi-urban and the rural population of nearly the low socioeconomic strata. And we had our women who, this is a cross-sectional study design where we had recruited women either delivering early preterm, late preterm, or term. Most of these women were asymptomatic, and the known causes of preterm births were excluded. The higher vaginal swabs were collected and they were subjected to microbial analysis, to 16S microbiome sequencing, and eventually some further PCR-based testing. And I'll share you the data which we have derived from these studies. So first and first, we looked at the microbiome. We did the 16S rRNA sequencing method, and we were hoping to see the vaginal dysbiosis. But when we looked at the total alpha diversity, there was no major difference between the full term and the preterm group. Also, with the beta diversity, we were hoping that these women would fall into two different PCAs, but in the PCA plot, as we see, most of the microbiome did strongly overlap between the women who delivered preterm and term. Even when we looked at the top 15 species at the taxa level, we did not find major difference between the species that were colonizing the vagina of women who delivered full term or preterm. We applied a number of tools, but in a sense, the end point was that there was no major difference in the vaginal microbiome of women who delivered preterm. We also did a subgroup analysis to look at the lactobacillus species more closely, and we looked at a number of them, but their abundance was not different between the women who delivered preterm and term. So the conclusion was that the 16S rRNA-based microbiome profiling did not explain the risk of preterm birth in our cohort. Now, we know that the 16S microbiome is great for global profiling, but if there are specific individual pathogens which may be associated with preterm birth, they cannot be predicted using, they cannot be identified using this tool. So towards then, we thought that, okay, let's look at specific pathogens which are known to colonize the vagina, leading to preterm birth, and the number of them which have been identified. Of them, we selected some of them, and we looked at whether these are found more commonly in preterm birth in India. Now, before that, we had conducted a systematic review, and we knew already from global studies that the frequency of group B streptococcus in women delivering preterm is between 18 to 20%. And in India, we did not know these numbers, so we conducted a systematic review where we collected all the published data of that we reported prevalence of preterm, prevalence of GBS in pregnant Indian women. And we had got data of about 9,700 women, and the results reveal that nearly the prevalence of GBS in pregnant Indian women is around 7.8%. And if we classify them by preterm and term deliveries, the odds of delivering preterm was fairly high when the women delivered preterm, and so was the odds of premature rupture of membrane. So this told us that yes, preterm birth and GBS are associated in the pregnant Indian women. But when we looked at the actual numbers, while the global numbers varied between 15 to 22% in India, we were getting less than half, and we were wondering why this is happening, what could be the cause. And then we analyzed the data further, and we found that the method of culture was one of the determinants of the prevalence of preterm birth. So, for example, on the prevalence of group B streptococcus. So, for example, those studies which had not done an enrichment step had reported a lower prevalence of GBS as compared to those who had used an enrichment step. So then Abhishek said that, okay, let's look at our cohort where we use a very consistent method. And by the time the carrot broth method was available, which is a very simple method where you take the swabs, put them in the carrot broth, incubate for 24 to 48 hours, and the change of color can determine the presence of GBS. And using this, we found that nearly 10% of the preterm birth women were positive for carrot broth, but also about 7.5% of the women who delivered full term were also positive for carrot broth. And we were wondering what's happening. And Abhishek sent these colonies for sequencing, and to our plight, we observed that none of these so-called GBS positivity were actually GBS. All of these bacteria which were isolated from these carrot broth, which were positive for so-called GBS, actually had sequence similarity to Enterococcus faecalis, telling us that what in the carrot broth positivity, what we were detecting was actually not GBS, but Enterococcus faecalis. And we reported this findings where we think that there's a false positive detection of GBS using the chromogenic media because of presence of Enterococcus faecalis in high vaginal swabs. And therefore, for all the microbiology community, it's important to remember that whenever we detect the presence of an organism, it's better to validate it with orthologous molecular methods to ensure that what we are reporting is what we are actually, or what we are detecting is actually what we are reporting, and vice versa. Anyways, by this time we said that, okay, the microbiology has its own limitations and we should go to the molecular methods. And by this time, WHO has already recommended use of multiple molecular methods for diagnosis of common infections, including bacteria. For example, in tuberculosis, the PCR-based test is well acceptable for diagnostic purposes. So towards that, Athara joined the lab, and he identified about 7 organisms which are strongly associated with preterm births, and he developed 2 sets of multiplex PCR assays which can detect about all of these 7 organisms along with lactobacillus thinners as a positive control in just 2 PCR reactions. And when we ran these PCRs on our cohort, we found a very different picture. These are now yes or no PCRs, that is presence of organism present or organism absent. And we found that both Gardnerella vaginalis and Streptogbs, we did not find any difference in their prevalence between the preterm and full term. In fact, we found that the GBS was in fact less prevalent in preterm as compared to full term. And we were wondering what is happening. However, some of the organisms like Klebsiella or Ureaplasma urolyticum were only found in women who delivered preterm birth. So we said that, okay, the PCR-based yes-no test is good enough for detecting some of the organisms and predicting risk of preterm births, but it is not completely helping us to identify more patients who do have these organisms but are not delivering preterm. So then we were wondering why this is happening, and we suspected that it is possible that while both the groups have this organism, the load of the organism may be higher in women who are delivering preterm as compared to term. That means that both of these have the organism colonization, but in one case it is maybe at a higher abundance as compared to other, and this could be the reason for such a phenomenon. So to do that, Sarika optimized something called disruptive digital PCR, which is not only telling us the presence or absence of the organism, but also telling us its copy numbers or its load. And when we applied this to our cohort, we found that Enterococcus faecalis, there was no difference in the load of Enterococcus faecalis in the higher vaginal swabs, but GBS was more abundant in the higher vaginal swabs of women who had delivered preterm as compared to term. However, while the levels were significantly different, only a small fraction of women who had preterm births had a higher abundance of GBS, telling us that although these levels may be significantly different, but in a larger context, they are not able to predict preterm birth with a higher accuracy. So then we moved to other organisms. We optimized these PCRs, and we found that the lactobacillus inverse copy numbers did not differ between the three groups. Neither was Klebsiella pneumoniae. Gardnerella vaginalis was found at a slightly higher abundance in the women who delivered early preterm as compared to full term or late preterm. The most interesting data actually came out from this other organism, which I'm going to not name it because we are awaiting to file the patent. But the data is very exciting. And here what we see is in our discovery cohort, we found that the abundance of this microorganism X was significantly higher in many of the women who delivered preterm as compared to full term or late preterms. Getting excited by this data, this is in a smaller study. So we also ran a validation cohort, and we found exactly the same result, that the abundance of the organism X was higher in women who delivered early preterm as compared to those who delivered term or late preterm, and the AUC was fairly high. And this had a very high specificity. That is, its ability to, that based on the cutoff that we had predicted from our AUCs, if we use that cutoff, we could predict the ability of delivering preterm with a fairly high positive predictive value, although the negative predictive value is not high, and this is anticipated because preterm birth is a heterogeneous condition, and in a number of them, it could be other causes, but not a high load of the organism X. But whenever there was a high load of organism X, we showed that the risk ratio is 2.6, telling us that if we use this as a tool, we should be able to predict the occurrence of preterm birth with a good accuracy. However, the rider here is that this information is derived from a smaller cohort. It requires validation in a longitudinal and multicentric setup. But if proven, we are very positive about it, that it should help in predicting preterm birth at a better accuracy and helping our clinicians and our women to manage these group of patients. So to summarize, in our cohort, vaginal dysbiosis is not strongly associated with preterm birth, but quantitative differences in the pathogen levels in the vaginal swabs of women who deliver preterm, and the quantification of such organisms may be able to predict early preterms with reasonable sensitivities and specificities. And if this works out, we are sure that it will help in developing specific intervention for personalized medicine to manage preterm births and neonatal infections and eventually reduce its burden in India. I thank my students and my staff, and also my clinical collaborators from the MGM Institute and Navreji Maternity Hospital, the Biostatistics Department of the Institute for helping us in some of the analysis, and the financial support provided by ICMR. Thank you very much. And I look forward for future collaborations. If anybody's interested in preterm births or also in other causes of infertility, we are looking for collaborators, and it is together that only we can do good science in this country. Thank you very much.